Hello, everybody. Welcome to my virtual talk. My name is Vyom ISBI. I'm a student at the University of Texas at Dallas, and I work at the Deep Learning for Precision Health Lab under Dr. Albert Montillo. And today I'll present improved motion correction for functional MRI using an omnibus regression model. A brief outline of the talk. I'll first go over the problem, what the impact of head motion on fMRI is, how previous approaches have tried to reduce head motion artifact, and what a fundamental problem is with previous approaches. And I'll present a possible solution where I'll go over what the new proposed omnibus regression approach is, how the efficacy of this approach compared to previous approaches is measured, and whether omnibus regression is better than previous approaches. So head motion is a significant source of noise in fMRI. For instance, in this plot, we have a carpet plot of a subject's image, and we can see the frame-wise displacement in red, which is a measure of the level of head motion at a particular time point. And on the y-axis for the carpet plot are the voxel intensities for a particular time point, flattened out into columns. And you can see that there are huge signal disruptions corresponding to the spikes in head motion. So head motion can actually account for 30 to 90% of the fMRI signal, in extreme cases over 90%. And they can cause distance-dependent artifacts in functional connectivity. This is because head motion can have three major types of effects on functional connectivity. They can inflate local correlations, they can inflate global correlations, and they can disrupt both global and local correlations. So the impact of head motion on functional connectivity can vary as a function of the distance between nodes in the brain. And finally, head motion can act as a major confounder. It can systematically affect data from children, elderly, or diseased subjects who move more compared to healthy controls in the scanner. And so removing this confound is imperative for valid analyses. So I want to emphasize that removing motion artifact is challenging. And in fact, there are nearly as many unique analysis pipelines in the literature as there are studies. In general, the pipelines involve a sequence of pre-processing steps, and I'll focus on the ones that remove head motion artifact. So removing head motion artifact itself involves a sequence of regression steps where artifact is removed by a linear regression of the data on nuisance covariates. So what does this actually mean? Well, this means that the data is fit to nuisance covariates using this equation, y equals x beta plus e, the generalized linear model. Here, y is the time series of intensity values for a particular voxel. X has columns that are time series of nuisance covariates, for example, head translation in the x direction and y direction and z direction and so on. Beta is the vector of coefficients obtained from the fit, and E is the residual denoised time series that is used for further analysis. So the problem is that a sequence of linear regression operations can reintroduce artifacts. This is because we can understand regression as a projection onto a subspace. For instance, here, X1 is one set of nuisance regressors that spans this blue plane, and projecting the data onto this subspace using this GLM gives us an orthogonal residual, which is E1, which will then be used for further analysis. If a subsequent linear regression is run on another set of nuisance regressors X2, it is highly likely that orthogonality with the previous subspace will be lost. So here we can see that the red residual is no longer orthogonal to the blue subspace. So noise is reintroduced from the X1 set of regressors. So the goal of our work was to create an omnibus regression model that combines the best artifact suppression algorithms and avoids reintroduction of artifacts from sequential regression, and to quantitatively evaluate this model against commonly used pipelines on a relevant data set. We chose a data set with both diseased and non-diseased subjects to capture a diversity of motion artifact. Specifically, we included 151 subjects from the Parkinson's Progression Markers Initiative, and these are the acquisition parameters. And here's a plot of the quantity of subjects in different cohorts. So PD is Parkinson's disease, prodromal and SWED are different phenotypes of the disease, and there's also healthy control subjects. Again, all of these are included to capture a diversity of artifact. We chose to consider 3 sets of nuisance regressors, with the first being the head motion parameters. These are obtained from the affine realignment of the data, and it gives it gives translation in X, Y, and Z, and rotation about X, Y, and Z. And we also compute square and derivative terms for a total of 24 nuisance regressors for the XHMP matrix. We also use the ICA aroma algorithm, which computes an independence components analysis on the image and classifies each component as either noise or non-noise. And we collected the noise components into another matrix called X aroma. And the number of noise components varied from subject to subject. And finally, we considered non-neuronal sources of physiological variance in bold by using eroded white matter and CSF masks and getting the average time series from those masks. Using these sets of regressors, we constructed 4 pipelines for our comparison. The first was a baseline where no motion correction was applied. The second was a sequential regression where first XHMP is removed, followed by aroma, and then physio. The third was a different order of sequential regression, where first aroma is removed, followed by HMP, and then physio. And finally, our proposed omnibus model, where we concatenate the XHMP, aroma, and physio regressors into an omnibus matrix and regress it at once. And this can be considered a fair comparison because the same set of regressors, the same class of regressors is used across these 3 pipelines. For our preprocessing, we followed standard steps for fMRI analysis, which include affine realignment, skull stripping, spatial normalization, smoothing, and the specific motion correction model or pipeline from the 4 mentioned before, and construction of functional connectivity matrices, which was done using a Pearson's R correlation between regional time series, and the images were divided into 333 regions using the Gordon Atlas. So how can the efficacy of these 4 pipelines be measured? Well, we focused on 2 quality control or QC metrics as they have been used and validated extensively. To compute these metrics, we need to quantify each subject's head motion level. And this was done using frame-wise displacement. Frame-wise displacement at each point is defined as a sum of the absolute values of differences in movements. So dx, dy, and dz are the displacement in x, y, and z. And theta x, theta y, and theta z are rotation about x, y, and z. So at each time point, this gives us a measure of how much motion there was compared to the previous time point. So our first QC metric was QCFC correlation, where a Pearson's correlation is computed between the mean frame-wise displacement, so the average level of motion for each subject, and the functional connectivity value for a particular edge across subjects. And this is done for all edges in the functional connectivity matrix. This gives us a measure of how contaminated or how associated the functional connectivity values are with head motion. And Ideally, this would be close to zero or zero, which would indicate there is no relationship between head motion and functional connectivity. Our second QC metric was QC-FC distance dependence. This is computed as a Spearman's rank correlation between the QC-FC value, so the previously computed Pearson's R correlation value for each edge, and the physical distance or the internode distance for that edge. Spearman's rank correlation is used to better account for non-linearities in this type of QC metric. And this gives us a measure of how associated are the internode distances and the motion contamination for each particular FC edge. And again, we want to ideally reduce this to zero, which would indicate no distance-dependent artifact. So our results, first with the QC-FC values, on the x-axis we have the 4 pipelines, and on the y-axis we have the absolute Pearson's R, which again we want to reduce to or bring to zero as close as possible. For the baseline pipeline with no motion correction, you can see that there is a fairly high correlation, median correlation between FC strength and mean frame-wise displacement, or so there's a strong association between head motion and functional connectivity. And in the comparison pipelines between sequential and omnibus models, the green pipeline with aroma followed by HMP followed by physio actually had the best performance, but it was fairly similar to the other 3 pipelines in that in the other 3 motion correction pipelines, in that none of them were able to reduce it to zero, and the median values had fairly similar values. So there's no clear winner here in terms of removing this artifact completely. For our second QC metric, again on the x-axis we have the 4 pipelines, and on the y-axis is the absolute Spearman's row. Each bar plot is annotated with the p-value for this association. So this p-value indicates the significance of the relationship between internode distance and motion contamination for a particular FC edge. And you can see the baseline without any motion correction, it's a very significant relationship. There's a fairly high association between distance and internode distance and motion contamination. And although the sequential regression pipelines were able to reduce this association significantly, the proposed omnibus model was the only one that could eliminate it completely and achieve a non-significant value for the association between for this distance-dependent artifact. So motion, as we've seen from these results, motion correction is essential, because without it, the baseline images and the derived functional connectivity measures are heavily contaminated. Further, the omnibus regression model removed distance-dependent artifact completely, and this was the only model in our comparison to do so successfully. And again, this is likely because sequential regression pipelines reintroduced noise that was removed previously. And finally, no pipeline could completely remove motion artifact, as seen by the QCFC measures. But that does not mean that that we cannot successfully reduce motion artifact, as successfully significantly reduce motion artifact. A major limitation of our study is that although our dataset was fairly large and diverse, it was only a single dataset, and replication on an independent dataset would validate our findings. Also, there is no absolute ground truth available with real-world data like this, so there's no true FC matrix against which we can compare the performance of motion correction pipelines. and simulation experiments could address this. Nevertheless, we still have proxies for how contaminated images are, or FC matrices are, by head motion. In conclusion, we proposed a new omnibus motion correction pipeline, which was able to significantly reduce and, in fact, eliminate distance-dependent artifact compared to the other pipelines, which were not able to do this. This omnibus pipeline can be used to directly reduce motion confounds in fMRI analyses and could improve the detection of true discriminative biomarkers for future analyses. And this proposed approach is disease agnostic and should be applicable to a wide variety of conditions where data is corrupted by motion due to different disorders. Thank you so much for attending my virtual talk. I would like to acknowledge my fellow lab members, especially Kevin Yen, who worked closely with me on this, as well as my PI, Dr. Albert Montillo. These are my funding sources, and if you have any questions or comments, please feel free to email me at this email. Thank you.